So we've been able to use a unique pancreas bioresource from individuals with type 1 diabetes to show that there appears to be two forms of the disease, and this is different in individuals diagnosed younger, under the age of 7, compared to those over the age of 13.
We've given those two different names. Different kinds of diseases are often called endotypes, and so we've used that word and we've called them type 1 diabetes endotype 1, which you can call TIDE 1, or type 1 diabetes endotype 2 in those who are older, or TIDE 2.
In the younger group, we find a more aggressive immune attack on the cells that produce insulin compared to those that are diagnosed older. And we also find that the insulin that is made in the beta cells is processed, we think, slightly differently in those individuals, and that might make them more susceptible to the immune attack. In the older group, we find that they actually keep many more of their beta cells, the ones that produce insulin, than we originally thought. And it may be that we can find ways to make these work better in the future, and hopefully that will lead to better, more effective treatments for those individuals.
What we've been able to do in this work is to study pancreas samples coming from children who very sadly died soon after they got type 1 diabetes. We've then been able to collaborate with our clinical colleagues to take blood samples from young people of different ages and show that what we find in the pancreas is mirrored in the blood. So it may be possible in the future to use blood samples to tell which kind of diabetes they have. This is really important because in the future it will allow us to target therapy in a very unique way to individual patients to stop them getting type 1 diabetes. That's never been possible before.